I'm Maike de la Roche. Welcome to my lab at the CRUK Cambridge Institute. We are interested in how the cells of the immune system communicate between each other and with the environment in order to mount powerful immune responses against cancer. The signaling pathway that we are most interested in is the hedgehog signaling pathway that is well studied in the fruit fly and during early embryonic development. But we have found that it has very important roles in the immune cells and can promote successful immune responses. In essence, my lab has two main interests. The first one is to understand the mechanistic underpinnings of hedgehog signaling in immune cells. And the second one is to translate this knowledge into designing novel immunotherapeutics for the clinic.
I study B cells, which is a type of immune cell that produces antibodies and helps protect us against infections and cancer. In particular, I am investigating how a pathway called the hedgehog signaling affects B cell behavior, how they mature, activate, and respond in the body.
So growing up in Nigeria, I witnessed the devastating impact of cancer firsthand. I've lost loved ones to the disease, and I also saw how the lack of access to personalised therapies like immunotherapy could further deepen the burden. And this inspired me to explore how the immune system interacts with cancer, with the hope that one day my research could help make the immune system a stronger and smarter ally in the fight against cancer and other infections.
I'm very, very lucky that I'm now in Maike's lab doing a very exciting project on immunotherapies where I develop novel therapies using people's immune cells to kill their cancers and hopefully benefit patients one day. So we are taking blood from healthy donors, from patients. And in the lab here, I take the blood and modify it to generate really, really good killer cells that we can give back to the patient. And the hope is that these killer cells, once they are back in the patient, are able to find the tumor and eradicate it, and thereby giving the life back to the patient and also giving a lot of life quality back to the patient.
I am interested in how the key signal transducer in T cells regulates their migration, and specifically using these insights to translate them into developing T cells that better migrate into the tumor and therefore better kill it. The research of T cell migration is essentially studying one aspect of T cell function, and everybody in the lab tries to study T cells or other types of immune cells from all sorts of different angles to really try to develop different approaches to power up the immune system to fight cancer.
What I'm interested in is how T cells can go into the tumor microenvironment and kill the tumor cells. The amount of T cells that can be found in the tumor is a really good indicator on cancer prognosis. And the more T cells you have, the more likely you are to be able to either reject the tumor or for the tumor to develop slower. I wanted to explore the role of hedgehog in the T cells found in the tumor microenvironment. And what I discovered is that the hedgehog pathway is actually really important for the migration of T cells into the tumors.
My research focuses on trying to understand why T cells become dysfunction in the tumors. And this is actually a very big problem in the clinic because these T cells, they should be killing tumor cells, and they can't because of the constraints of the tumor microenvironment. We are interested in trying to understand the role of hedgehog on T cells, but we don't know what's the role of hedgehog signaling in these dysfunctional cells. So my work is focusing on that, which is more of a fundamental type of question, but the impact of solving these problems can have implications, for example, for CAR T cell therapy.
My research focuses on developing new specific treatments against cancer cells without harming healthy cells in the body. I am particularly interested in cancers that are currently hard to treat. I am trying to develop nanoparticle-based treatments as well as bispecific engagers that drive the immune system of the patient against the cancer, thus limiting the standard side effects that are associated with chemotherapy. Coming from a clinical background, I have firsthand experience of the frustration patients experience when they are out of options, and I chose to go into basic research to make an impact and contribute to solving this problem.
Understanding the basics of hedgehog biology and immune cells enables us to uncover novel avenues to design and make immunotherapeutics. One crucial aspect of our work is our close collaboration with clinicians and patients. Using human samples, we can direct our research efforts to the most promising therapeutic candidates and thereby delivering impact where it matters.